Assalamu alaikum. Today we want to talk about chapter 20, the generation of biochemical energy. And we will cover inshallah in two parts the following topics as time allows. Now, energy and life. Living things do mechanical and chemical work, synthesizing molecules and moving them across cell membranes. Now, the energy used by all but a very few living things on Earth comes from the sun. Plants convert sunlight to potential energy stored in the bonds of carbohydrates. Animals use this energy and store the excess in the bonds of fats. As you can see here, plants take energy from the sun, convert it to chemical energy, then animal release of chemical energy in catabolism. They do work and they have heat. Now chemical synthesis, A plus B plus energy to give AB, and the combining of two molecules to give a larger one. And then we have transport across membrane. Then we have maintain body temperature plus loss to surroundings. Even the heat we get, some of it will be used to maintain our body temperature, and the majority of it is lost to the surrounding. So our bodies have specific requirements for energy. Energy must be released from food gradually. It must be stored in accessible forms. Release of energy must be finally controlled. Just enough energy must be released as heat to maintain constant body temperature, and energy must be available to drive chemical reactions that are not favorable at body temperature. Now, chemical reactions either release or absorb energy according to the formula. Delta G is the free energy change, delta H is the enthalpy of the reaction, minus T is the absolute temperature, and delta S is the change in the entropy of the system. Reactions in living organisms are no different from reactions in chemistry lab. Now, spontaneous reactions release free energy. Exergonic reactions are the source of biochemical energy. All combustion reactions are spontaneous, so they release energy, and we call them exergonic reactions. Here we have two types of energy diagram: free energy versus reaction progress. Here we have, this is an example of an exergonic reaction. This is a favorable reaction where delta G is negative, equal G of products minus G of reactants is negative. So the sign of delta G is indicative of exergonic reaction. On the other hand, here delta G equal G products minus G reactants. This is a positive quantity, so this is an endergonic reaction and it is an unfavorable reaction. Now in our body, the energy released from exergonic reactions drive endergonic one, and this is called coupling, and coupling is what metabolism is all about. The greater the amount of free energy released, the further a reaction proceeds toward product formation before reaching equilibrium. Now, reactions requiring an input of energy are endergonic. Free energy changes switch sign for the reverse of the reaction, but the value does not change. If we take, for example, this fixation of CO2 in the photosynthesis to form glucose, delta G is positive. This is an endergonic and energy required process, whereas the oxidation of glucose by oxygen to CO2 plus water, delta G sign is negative. You see, both the quantities are equal but with opposite signs, and this is an exergonic energy releasing reaction. Living systems make use of this in biochemical pathways. Energy is stored in the products of an overall endergonic reaction pathway. This stored energy is released in an overall exergonic reaction pathway that regenerates the original reactants. Again, What is an endergonic reaction? Is a non-spontaneous reaction or process that absorbs free energy and has a positive delta G. However, an exergonic reaction is a spontaneous reaction or process that releases free energy and has a negative delta G. So you see, the sign of delta G is indicative whether the process is endergonic or exergonic. And all exergonic reactions are spontaneous, whereas all endergonic reactions are non-spontaneous. There are two main categories of cells, prokaryotic and eukaryotic. Prokaryotic cells are usually found in single-celled organisms like bacteria, blue-green algae, etc. Eukaryotic cells are found in single-celled yeast and in all plants and animals. So we are considered eukaryotes. Eukaryotic cells are about 1,000 times larger than prokaryotic cells, and features include membrane enclosed nucleus. Organelles are small functional units that perform specific tasks. Then we have cytoplasm, is the region between the cell and the nuclear membrane. Cytosol is the fluid part of the cytoplasm with electrolytes, nutrients, and enzymes in solutions. And this is the shape of the structure of the cell. You see here we have the mitochondria and cytosol. These are the one important to us, where many processes occur in cytosol, others occur in mitochondrial matrix, others occur on the endoplasmic reticulum, and other protein synthesis occur on ribosomes, etc. And maybe fatty acid chain lengthening occur in Golgi apparatus and some mitochondria. And again, these are the cell component, and as I told you, what is the principal function of each of these components? For example, ribosome protein synthesis, mitochondria synthesis of ATP from ADP, cytosol intracellular fluid contains dissolved proteins and nutrients, etc. And don't worry about the cells and their structure, don't worry. Now we are concerned about mitochondria. The cell's power plants are the most important of the organelles for energy production. You see here where citric acid cycle occur in the matrix, in electron transport and oxidative phosphorylation, etc. Here ATP synthase enzyme sites of ATP production. ATP, CO2 comes out, ATP comes out from here. Glucose, O2, ADP, and phosphate enters here. This is the matrix. This is the mitochondrial matrix, inner membrane, outer membrane, and intermembrane space. This is a very important part of the cell, as you can see here. CO2 out, oxygen in, and so on. Now, the citric acid cycle takes place in the mitochondrial matrix. Electron transport and ATP production take place at the inner surface of the inner membrane. The numerous folds in the inner membrane, known as cristae, increase the surface area over which these pathways can take place. An overview of metabolism and energy production. Now, metabolism is the sum of all the chemical reactions that occur in our body, and most reactions occur in metabolic pathways. For example, we could have a linear sequence A to B to C with the three enzymes. We could have a cyclic sequence. We could here have a spiral sequence, again with a starting point, and then we have final product. And we will see examples of these as we go, because we are going to study the different pathways that lead to the breakdown of large molecules into smaller ones. Metabolism consists of two parts, catabolism and anabolism. Here in catabolism, metabolic reaction pathways that break down food molecules and release biochemical energy. So you break larger molecules to smaller ones. In anabolism, the opposite, metabolic reaction that build larger biological molecules from smaller one. Now, for example, catabolism is energy releasing, oxidative in nature. Large molecules are broken down to smaller one. Anabolism is reductive in nature, energy requiring, where small molecules are used to build larger ones. As you can see here, this is an example. So smaller molecules plus energy, anabolism to form larger molecules. Catabolism, larger molecules produce smaller one plus energy. So this is summary is illustrative. Now an overview of metabolism and energy production. Notice here we have food. We have three major types of biomolecules. Lipids, carbohydrates, proteins are broken down into amino acids, and carbohydrate to glucose and other sugars, lipids to fatty acids and glycerol. You see this will end up as a molecule called acetyl-CoA, as well as amino acid catabolism. Carbohydrate through glucose is converted to pyruvate, as we studied before, and then to acetyl-CoA. So we have these stages: digestion, where larger molecules are broken down to smaller one. Then acetyl-CoA production, sugar and amino acid molecules are degraded in the cytoplasm of cells to yield acetyl-CoA. Fatty acid molecules are degraded in the mitochondria of cells to yield acetyl-CoA. Then stage 3, the citric acid cycle. Now this acetyl-CoA is oxidized inside the mitochondria by this citric acid cycle to yield CO2 and reduced coenzymes, plus a direct ATP synthesis. Then ATP production, the energy transfer to the reduced coenzymes like NADH and FADH2, which we will see, are used to make ATP by the coupled pathways of electron transport and oxidative phosphorylation. Then let's start with stage 1, digestion. Enzymes in saliva, the stomach, and the small intestine convert larger molecules into smaller ones. Carbohydrates broken down to glucose and other sugars, proteins down to amino acids, and lipids are broken down to glycerol plus long-chain carboxylic acid and fatty acids. These smaller molecules are transferred into the blood for transport to cells throughout the body. Then stage 2, acetyl-CoA production. The small molecules from digestion follow pathways that move their carbon atoms into two carbon acetyl groups. This is acetyl-CoA, acetyl coenzyme A, and this is the acetyl group. The acetyl groups are attached to coenzyme A by a bond between the sulfur of the thiol group and on coenzyme A and the carbonyl carbon atom of the acetyl-CoA. We call this a thioester bond, and it is a high energy bond. Then the small molecules from digestion follow pathways that move their carbon atoms into two carbon acetyl group, which are attached to coenzyme A by a bond. Now acetyl-CoA is an intermediate in the metabolism of all food molecules, as we will see. Now stage 3, the citric acid cycle. Within mitochondria, the acetyl group carbon atoms are oxidized to the carbon dioxide that we exhale. Most of the energy released in the oxidation leaves the citric acid cycle in the chemical bonds of reduced coenzymes, NADH and FADH2. Now some energy leaves the cycle stored in the chemical bonds of ATP or a related triphosphate like GTP, and we call this substrate level phosphorylation, as we mentioned before. In stage 4, ATP production, electrons from the reduced coenzymes are passed from molecule to molecule down an electron transport chain. Their energy is harnessed to produce ATP. At the end of the process, these electrons, along with hydrogen ions from the reduced coenzymes, combine with oxygen to produce water. The reduced coenzymes are oxidized by atmospheric oxygen, remember, and the energy that they carried is stored in the chemical bonds of ATP molecules. Then ATP, even we cannot say no. ATP is a form of energy on demand. This is the structure of ATP: adenosine triphosphate, and adenine purine base. If we attach ribose sugar, it becomes adenine become adenosine, and then when we add the phosphate, it becomes adenosine triphosphate. Then a purine base plus ribose sugar nucleoside. Then when we add the phosphate, it becomes a nucleotide. And here we have extra groups now. Then this is the triphosphate group, and this is the bond broken in hydrolysis to ADP, and this hydrolysis will yield around 7.3 kilocalorie. Now, removal of one of these phosphate groups by hydrolysis gives adenosine diphosphate. As you can see, delta G is this amount. Now, if you want to reverse this process, the phosphorylation of ADP to ATP requires the input of 7.3 kilocalorie per mole. So ATP is an energy transporter because its production from ADP requires an input of energy that is then released wherever the reverse reaction occurs. So remember these two things. Strategies of metabolism: ATP and energy transfer, the hydrolysis of ATP to ADP and its reverse, the phosphorylation of ADP are reactions suited to their role in metabolism. The stored energy is released only in the presence of appropriate enzymes. طبعا. A useful amount of energy is released when a phosphoryl group is removed from it by hydrolysis. If too much energy was involved, interconversion would be more difficult. Now, strategies of metabolism, metabolic pathways and coupled reactions. The overall free energy change for any series of reactions can be found by summing up the free energy changes for the individual steps. Not every step in the metabolic pathway is downhill. Energetically unfavorable reactions are coupled to energetically, as I said, coupling is what metabolism is all about. This coupling allows the energy stored in the chemical compound be transferred to other compounds, where excess energy is released as heat and contributes to maintaining body temperature. Here we have metabolic pathways and coupled reaction. Example, we have this compound called phosphoenol pyruvate. The hydrolysis of this bond, delta G, is minus 14.8 kilocalorie. However, the phosphorylation of ADP to ATP requires 7.3. So if you add these two together, you get the difference between them. So phosphoenol pyruvate plus ADP بتعطينا pyruvate plus ATP, and the sum. Then for coupling to occur, there must exist common intermediates. As you can see, water, and inorganic phosphate, they cancel out. And also the sum of their delta G must be negative. As you can see, if we add this to this, we will get a negative quantity. So coupling is possible here, and the result is negative.